Hi, everyone. Okay, this is the final lecture on Mendelian genetics, or just genetics, where we actually cover non-Mendelian genetics. Beginning with incomplete dominance, we will compare that to codominance, then compare pleiotropy to polygenic inheritance, and then we'll wrap it up with sex-linked genetics. All right, so before we begin, I know that you've seen these lectures already in class, but you should think of these going forward as maybe blocks where those two should be compared, incomplete dominance with codominance. And then you should also compare pleiotropy with polygenic inheritance. Those kind of go together. And sex linked is the one that's kind of um, standing apart from all the others. So let's go into a comparison of incomplete dominance with codominance and how that's different from Mendelian genetics, okay? So if you remember in Mendelian genetics, there's one gene. That one gene has two alleles. We can also look at two genes, three genes, all at the same time. But in each case, there's two alleles, and one allele dominates over the other. And for that reason, we get some typical genotypic and phenotypic ratios, right? The ones from the cheat sheet that we may have covered in a previous lecture. But in the case of non-Mendelian genetics, the crosses produce genotypic and phenotypic ratios that are different. So let's look at examples of those. In incomplete dominance, none of the two alleles will dominate. Already that's a departure from Mendelian genetics where one allele dominates over the other. So when neither of the two alleles dominates, you're going to get some sort of intermediate phenotype. You should think of this as like a blending scenario, like mixing paint. Cross a red with a white, whatever, or mix red paint with white paint, and you'll get pink, right? In Mendelian genetics, if you mixed the red with a white, you would have gotten all red or all white. This is not that case. Examples in nature would include snapdragons, blue Andalusians, a genetic condition in humans called hypercholesterolemia, and then in the case of another mammal, the palomino horse. Um, so here's examples of snapdragons and carnations, right? These would be the carnations over here. And over here would be the snapdragons. And you can see that there's clear reds, clear whites, and here's a pink. That's the product of crossing a red with a white. We'll do a cross in a second to show that. And down below, you're looking at a carnation that is red, one that is white, and if you were to cross those two, you'd get this pink one in the middle. All right, so it's very much like blending, okay? So let's take a look at some crosses of, let's say, carnations. So in the case of carnations, we know that if you are red, you're homozygous for red. That would be like big R, big R. Notice that there's no lowercase letter because there is no recessive. And if you were to be white as a carnation, or if you're a carnation that produces white flowers, you would be R prime, R prime. And finally, if you were pink, you'd be R, R prime. So notice that these two would be considered homozygous, right? They have two of the same alleles, big R, R prime, R prime. And this is the only one that's heterozygous, right? Okay, so in this case, if you were to cross, let's say, a pink with a pink, which is hetero with hetero, Mendelian genetics would have predicted that you get a 1 to 2 to 1 genotype ratio and a 3 to 1 phenotype ratio. Let's see if that's true here. So we're going to cross pink with pink. Why wouldn't you get more pink, right? So there's R, R prime, crossed with R, R prime. Set up our Punnett square, put our alleles in the Punnett square, and then start bringing those alleles together. All right, now that we've filled it out, now we pull out the information. Let's start out with the genotype ratio. What do I see in this first box? I see big R, big R. There's one of those. I also see big R prime. There's two of those. And I see R prime, R prime. There's one of those. All right, so far it's looking like Mendelian genetics. That's a 1 to 2 to 1 phenotype ratio. But let's look now at the phenotype ratio. I see in here a red. There's one of those. I see two that are going to be pink. And I see one that's going to be white. Okay, Mendel predicted a 3 to 1 ratio, and here I'm getting a 1 to 2 to 1 ratio, just like I did the genotypes. All right, so that's totally different from Mendelian genetics. And also, of course, the fact that there isn't a clear dominant and a clear recessive. So let's go back to those flowers. Look at the red carnation and the white carnation. What do we know about those whenever we see them in nature? They're homozygous. They carry two of the same alleles, two reds or two whites. And whenever we see a pink carnation, now you know that those are heterozygous. It has one white allele and one red allele. All right, other examples of incomplete dominance include hypercholesterolemia. Notice that the homozygous normal is homozygous. Notice that the individual with the extreme case or the severe case of the disorder is also homozygous. But then notice that the individual that is with a mild case is hetero. All right, so what explains incomplete dominance? How is that different from the dominance and recessiveness that we learned about in genetics, where if one allele is functional, it's producing an enzyme that works. If the other allele is not functional, it's producing an enzyme that doesn't work. And because enzymes are reusable, as long as one of the enzymes works, it makes up for the fact that the other enzyme doesn't work. So again, you could be heterozygous for earlobes, and this gene doesn't work. So you're going to make an enzyme that doesn't work. But because this one does work, you'll make an enzyme that does work, and it'll make up for that one because enzymes are reusable. So as long as you got one functional copy, it dominates. But that's different here in incomplete dominance. Why is that? Well, here in this case, you have two chromosomes, right? One has a big H, the other one might have a small h. But if you have a big H, that one is going to be good enough to make a certain amount of receptors. If both of them were working, then you'd make the other half of the receptors. So having this chromosome is going to make those two receptors. Having that chromosome that also works will get you the other two receptors, and now you will have the full amount of receptors. But what happens if you have two genes that don't work? Well, you make 0 receptors. And what happens if you have one that works and one that doesn't? Well, this one works, so you'll get the 2 that it's supposed to produce, but the other guy makes no other contributions, and so now you have half of the amount. Because you don't have the full amount, you don't bring in cholesterol the way normal individuals do. You would have a mild form of the disease. So this is still the story of proteins, but these proteins are not enzymes. They're not reusable. These proteins, right, are receptors here. You need to have the full amount of receptors to bring in your cholesterol efficiently. Otherwise, it's inefficient, mild disease. And worst off, if you don't produce any of the receptors, you have a severe case. You'd have to have modified diets where you limit your cholesterol intake. All right, same can be said about the blue end dilution. If you have two fully functional chromosomes, you'll get the full pigmentation, you'll be black. If you don't, you'll be white, and if you get one that works and one that doesn't, you'll show up as gray. Breeders are notorious for calling gray things blue. So this one's the heterozygous, the blue one or the gray one. Same is true for the palomino. Full production of pigmentation, none, a little bit of. All right, so those are examples of incomplete dominance where you get blending, okay? And we did a Punnett square to show that. Let's go back in time. All right, what's up with codominance then? All right, well, that's different. In this case, both alleles will dominate, and for that, let's go over blood types. All right, in blood types, we generally focus on the blood types that are important in the clinical setting, right? These are the blood types that we all learn about. You should know your blood type, and if you don't, at your next opportunity, you should get that information. So we're gonna focus on the ABO blood groups, right? And A, B, and O, those are 3 alleles. Already, that's different from Mendel, where one gene had 2 alleles. Here you have one gene with 3 alleles. So that's going to be a departure of Mendelian genetics, and it's going to give us totally different outcomes. So if we were looking at the phenotypes that people can have, they can have a A-type blood, they can be blood type B, they can be blood type AB, they can be blood type O. And we've left off the Rh antigen, the negative or the positive. That's a separate gene. That would be like a dihybrid cross. You can look at the gene that controls what sugar you put on, A or B or no sugar. And then you can look at another gene that decides whether you make a protein or not. If you do, you're positive. If you don't, you're negative. This is the example of codominance right here. Whenever you say that a person is blood type AB, we're saying that they're both A and B. That's co-dominating. Both alleles are showing up on that individual. So let's do an example of blood types, or maybe 2 examples. Again, in this case, you can list the blood type, and then we should know the genotype. That's going to be important. So you can be blood type A, in which case your genotype can be homozygous for A, or you can be heterozygous for A. Your blood type B, you can be homozygous for B, or you can be heterozygous for B. You can be blood type AB, in which case you would have one of each. That's the only way to be AB. Or you could be blood type O, in which case you're little I, meaning that you're not producing any sugar on the surface of your cells. So your white blood cells that you saw in lab can either have an A-type sugar, or they can have a B-type sugar. But individuals that are AB, they have both. They have both A and B sugars on the surface of their white blood cells. Okay, so let's do a cross. Let's say we had an individual that was blood type AB, and we crossed it with another individual that was also, let's say, blood type AB. What kind of offspring would they have? All right, this is hetero with hetero. So we might be expecting like 1 to 2 to 1 genotype ratios, 1 to 2 to 1 phenotype ratios. Or I'm sorry, 3 to 1 phenotype ratios. All right, so this person would have, or these 2 people would have potentially a child that's blood type A, homozygous for a child that is AB, another child that's also AB, or a child that's blood type B, homozygous for B. So it looks to me like the phenotype and the genotype ratios would be 1 to 2 to 1, both of them. Just like what we saw in incomplete dominance. So your genotype ratio, that's an ugly G. Your genotype ratio would be, you get a big A, there's one of those, big A, big B, there's two of those, and finally big B, there's one of those. There's your 1 to 2 to 1. And your phenotype would be, all right, there's the blood type A, AB, and B. That's also a 1 to 2 to 1 phenotype ratio. And it gets really interesting if you cross like heteros with heteros, like those two heteros, right? Wouldn't you say that these are heteros also? Remember, the definition of a heterozygous is having two different alleles. So these are also heteros. There's like multiple ways to be heterozygous here. But what happens if you cross hetero with hetero of this kind? You'll get some really interesting genotype and phenotype ratios. So let's do that cross really quickly. And then you might want to take me up on my offer to try some of the genetics problems that I uploaded on Canvas. Maybe work those out with a classmate. Talk with each other. Do bio with each other. Learn from each other. Okay, so I just finished that Punnett square. What's my genotype ratio for that cross? Well, if I look in here, I see that there's a big A, big B. There's one of those. I can also see that there's a one of those. I can also see that there's one of those, and I can see that there's one of those. Okay, that's a 1 to 1 to 1. Phenotype and genotype ratio. They're all different blood types, and they all have different genotypes. So the genotype and the phenotype ratio are the same, 1 to 1 to 1 to 1. All right, you are going to see this kind of genetics problem on exam 3. You need to make sure that you know this. If I say blood type A, you need to know that that could be homozygous, or I should extend it over, or you can be heterozygous. You got to know that. If I say blood type B, you got to know you can be homozygous or hetero. All right, let's practice that. This slide is from a time when we used to do blood typing in the lab. We don't do it anymore, so it's kind of losing its value, but maybe I'm holding on to it because I'm nostalgic for the olden days as I'm getting older. Okay, here's another example of having multiple alleles, right? Blood types are an example of multiple alleles, A, B, O. In rabbits, their coat color can also show multiple alleles, right? You can see all the alleles right there, right? C, capital C, lowercase c with an H, lowercase c with a CH, right? There's multiple alleles there. And because there's multiple alleles, you get a wide array of phenotypes. The more alleles you have, the more combinations you can have, the more different phenotypes you can have. All right, let's compare now pleiotropy with polygenic inheritance. So in this case, you have one gene that gives you multiple phenotypes. Or in polygenic inheritance, you can have multiple genes that give you one outcome, right? So I tend to visualize a pyramid when I compare these two. Maybe you're like, I don't know what this pyramid's all about. All right, forget I ever said anything about pyramids. They don't work for you. But for me, it's visually, it kind of makes sense. All right, so one gene giving you many different phenotypes, that would be like genetic disorders. A lot of genetic disorders are like that. Metabolic disorders usually are the kind of disorders that we have. And that gene gives you multiple symptoms, and each one of those symptoms is a phenotype. So let's look at sickle cell anemia, one gene, and look at all of the symptoms. Every one of these boxes is like a phenotype, so physical outward appearance. one gene, multiple phenotypes, as opposed to earlobes, one gene, you're either free or you have attached earlobes. Here, there's like way more other outcomes. Sickle cell anemia is a condition seen in among our African American uh, members of our population, whereas the Caucasian members of our population would very likely have cystic fibrosis, which is an excessive production of mucus that leads to all kinds of respiratory issues and other um, like organ impairment issues. And so you would have multiple symptoms associated with that. Also, an example of pleiotropy. Um, PKU is another example. There's Tay Sachs. I mean, there's just a lot of genetic disorders lend themselves to be examples of pleiotropy. So on exam 3, if you're reading a question and it says something about a genetic disorder, immediately you should be thinking pleiotropy, right? And maybe you work out a problem or something like that. Okay, um, polygenic inheritance is exactly what the term implies. Poly, many, genic, genes. So many genes are involved here, and they give you one outcome. Skin tone is the example that we like to use, right? And you should know that skin tone is also an example of incomplete dominance. That's definitely on exam 3. So be prepared for that question. Okay, so here's an example of skin tone in a population graft, right? So look at the skin pigmentation among members of a population and the extent to which you would find darkly pigmented individual versus light pigmented individuals, and then all of this spectrum in between. So whenever there's a spectrum like this in nature for anything, not just humans, but we can look at humans because that's what we understand more of. Hair color, eye color, height, right? All of these are a spectrum. There's a variety, there's a wide range of outcomes. And for that reason, we're very likely um, seeing examples of many genes contributing to one outcome. So in this case, we're looking at a very dark pigmented individual and a very light pigmented individual, and they would have children that are intermediate pigmentation. Do you see the incomplete dominance example there? Dark, light, but the offspring are intermediate. That's a blending thing, so that's incomplete. That's why we say that. And if you had an intermediate pigmented individual marry an intermediate pigmented individual, notice that they can also have a very dark pigmented child, or they can have a very light pigmented child. And you can sort of see the potential of that happening there. You're seeing a fraction, so you're seeing chances. So there's a 1 in 64 chance that they will have a child that's darker than both of them, like significantly darker than both parents. A 1 in 64 chance that they can have a child that's significantly lighter than both parents. All right, so it does happen, right? Genetics can explain two parents having a very dark child or a very light child. Okay, by the way, you might also recognize this kind of question as if it's polygenic, right? That's where you do the law of probabilities, right? We would not do that Punnett square, correct? So maybe we should do another practice example of the law of probabilities. So we have this individual crossed with this individual. And again, you can do the A's by themselves. And you can ask, well, maybe we had a question like, what are the chances that they'll have a child that's darker than both of them? All right, so the chances of that happening seem to me to be 1 out of 4. And then we can do the B's separately. And we can can ask, okay, what are the chances that we'll have a child that's little b, little b? Looks like there's 1 out of 4. And then finally, we can do the C's. And we can ask, what are the chances that we will have a child that's big C, big C? Looks like that's also 1 out of 4. And if we do the math, that's 1 in 64. We could also be asked, okay, well, what are the chances that they'll have a child that is, I don't know, heterozygous there, but homozygous there, and also heterozygous there? Okay, well, let's just go back and look at the same Punnett squares. What are the chances of getting big A, little a? Looks like it's 1/2. What are the chances of getting little b, little b? Looks like that's 1/4. And what are the chances of getting big C, little c? Looks like that's 1/2, right? So we do our math. 1 times 1 times 1 is 1. 2 times 4 is 8. Times 2 is 16. Remember, don't add. Multiply. This is the law of multiplication. All right. I talked about this in lecture. I don't know that I need to talk about it here again, but this young couple made the news because they had fraternal twin daughters, and when you compare the two, like, they're clearly on opposite ends of the spectrum, or close enough to being on opposite ends of the spectrum, I would say. And it's just like an interesting outcome of nature in the context of genetics, right? All right, very cool family picture. We also talked about the lab as an example of having genes that control other genes, right? That's a big deal in genetics. So there's a gene that controls coat color. You can be, for example, big B, big B, big B, little b, in which case you'd have a black coat. Or you could be little b, in which case you'd have a brown coat. Those are the genotypes. However, there is another set of chromosomes that control whether you put down the pigments or not. You could be big E, or big E, little e, in which case you get to put down whatever that state. You can put either one of those down. In other words, you can put pigmentation down, black or brown. But you can also inherit little e, in which case you wouldn't be putting any pigmentation down. Doesn't matter what you have. If you got that, you're not putting any of it down. No black, no brown. You end up being a yellow lab, which is pretty cool. And so when we look at labs, we can see that this lab right here is clearly a black lab, so it has to have at least a big B, right? And because it's black, it must have at least a big E. Do we need to know what goes there? No. We know that you have at least one dominant allele making you black, and we know you have at least one dominant allele allowing you to put down that color. So it doesn't matter what you have here. You have a big B, you have a big E, that allows you to be black. What about for the chocolate one? Well, it has to be little b, little b. That's the only way to be chocolate. And you have to have at least one big E. Doesn't matter what goes there. We actually know more about this dog than we know about the black dog. And what about the yellow lab? Okay, well, the yellow lab is very definitely little e. But can we figure out what it has, black or brown? And what I say is that, well, if we can see that you have some red on your nose, red lip liner, red eyeliner. I'm sorry, what? I meant black. You have a black nose, black eyeliner, black lip liner. It's late. Then that must mean that you carry at least one black allele, and we don't know what the other allele is, but it doesn't matter. You've got at least one black allele. You could be carrying a little b, you could be carrying another big B, it doesn't matter. The fact that you're showing black means that you have at least one black allele. And I say that because notice that in the case of the chocolate, the nose is brown. There is no black on the lip liner. There's no black on the eyeliner. So this one's clearly little b, little b. No ability to make black because they don't carry it. All right, good stuff. Share that with your vet. Share that with people out in the street that have labs. It's a cool talk. It's a good way to meet people. All right. We should also ensure that you understand that the environment can have an effect on your genes. In your case of earlobes, your environment isn't going to do anything unless, of course, the environment is you like putting some of those, I don't know, what are they called? Like those like triangular things that stretch out your ear and make bigger and bigger holes in your ear. What's all that about? I'm showing my age or something. Um, yeah, I suppose that's an environmental thing doing something to your ears. I can't, but nature as an environment doesn't really impact the shape of your ears or anything like that. But we do know about um, exposure to sunlight having an effect on your skin tone, right? We know that things like heart disease and cancer can be influenced by your environment, like your diet, exposure to the sun itself, smoking, things like that. So your environment can definitely have an effect on your outward appearance by way of affecting your genes. So your genes and the environment are interacting with each other. You remember the case of the Himalayan rabbit. If you covered an ear or if you put it in an incubated room, a room that's warmer, it would go to being solid white, right? So your environment does have an effect on genes, or you are a product of your genes and the environment. It's not just all genetic. These hydrangeas are affected by the soil pH. They change color in one pH. They go to a different color in another pH. So the pH of the soil has an effect on the color of the hydrangeas. That's just another example. All right, we don't have time for Hellboy, but let's talk about sex. All right, so I think at this point we're all comfortable with this idea that males are going to carry an X and a Y chromosome, whereas females carry two X's. So when females go through meiosis and they separate the two sex chromosomes, all of their eggs are going to carry definitely an X. So they're all the same. They're all uniform. They carry X's. But when a male goes through meiosis, he can give either the X or he can give the Y chromosome. And that's basically why we say that males determine the sex of a child. Although there's, again, recent evidence by way of research that suggests that the female also has a say in, what kind of sperm fertilizes an egg, right? Although it doesn't mean that they decide male or female. The egg is actually deciding for sperm from a male that has greater genetic variety. So, if a female were to have mated with two males in recent time, and one of those males has more genetic variety than the other male, apparently the egg would opt for the gametes of the male with greater genetic variety. I'll put up something on Canvas maybe about a study on that. Let's look at the sexing of other insects or other species like insects. Notice here that there's 22 autosomes and one sex chromosome. So in their world, having one sex chromosome makes you a male, but if you had two sex chromosomes, you're a female. See the difference? And in birds and some other lizards, we have a total reversal of the sex system, right? It would be XX is a male, XY is a female. But we go with ZZZW. We adopt a totally different system to remind people that this is the opposite. Because if we write XX is a male and XY is a female, that's going to confuse everyone. So we're like, okay, maybe we don't bring those up. Let's just create something brand new, ZZZW. But the cool story for me is the bee. And I'm definitely putting that on exam 3. You should know that bees are haploid, and male bees are haploid, and female bees are diploid. Got to know that. So what's happening here is that a female puts down an egg. The egg has 16 chromosomes. And if the egg gets fertilized, it would go back to having 32 chromosomes. And in that case, you would be a female bee. However, if the egg with 16 chromosomes doesn't get fertilized, then it just develops into a male. So male bees are actually haploids. They're like multicellular eggs. And I think that's just awesome. All right, this is the calico cat. The calico cat appears to show us an example of codominance, right? The calico cat um, can have these genotypes. So the female can be X big B, in which case she's black. She can be X little b, let's say, in which case she's orange. Or she can be X big B, X little b, in which case she's calico, meaning she's both black and orange. You should recognize that as looking like codominance. In the case of males, they can be either black, for example. Oops, let me go back. In the case of males, they're XY, so they can be either black or they can be orange, but they cannot be calico, right? It's impossible because they don't carry two X's. They only carry one. The other one is a Y chromosome. So they cannot be calicos. Or can they? All right, there's a twist to this. All right, so in this case, the female here in this image is showing both black and orange. And again, that looks like codominance, right? But the thing happening here is that one of the two chromosomes, one of her two sex chromosomes is shut down so that she's only using the other one. In this case, the one that's producing orange right there. And then in other parts of her skin, she's shutting down the orange one so that she's only operating with the black chromosome. And so she's got black coat there. So we're getting the illusion of codominance, but in reality, it's something else. This is something called X inactivation. And what's interesting is this. There are rare cases where there's a male that is calico. So can you figure out how that's possible? All right, we said it's male, so it has to carry what chromosome? Y. And we said it's calico, so it can only be what? There. It's a Klinefelter cat. It's like the human condition, right? Where you have like non-disjunction, a syndrome, Klinefelter syndrome. There's cat versions of that. So whenever you come across a calico cat, pick it up, inspect the cat. It might be a male, and that cat is very valuable. They're rare cats. Cool. All right, let's do some crosses with sex-linked genes. So in this case, we're doing now sex-linked genetics. Sex-linked genetics focus on the X chromosome, so we call them instead X-linked. All right. We don't really focus on the Y chromosome, but I don't know, I can change that maybe going forward. Maybe, I mean, there are some interesting, weird genetic disorders associated with the Y chromosome, but they're rare. The X-linked disorders are more common, and they include the following, and you should document these. Color blindness. Is that with two S's? I'm going to add it just in case. Hemophilia. And Duchenne's muscular dystrophy. I think it's two E's. Two N's, I meant. Muscular dystrophy. This is the inability to develop your muscles. Hemophilia is an inability to clot. You're missing a clotting factor. So clotting is like a complex mechanism of multiple factors coming together to form a clot. People with hemophilia are missing one, and so they don't clot well. And then color blindness, I think, you know about, right? That's an inability to tell different colors apart. And those genes are found on X chromosomes, right? So let's go with color blindness. That's an easy one to track. In the world of humans, females can be XX with normal vision, so they would be homozygous for normal vision. Or they can be homozygous colorblind, or they can be heterozygous carriers, we would say. They carry the recessive allele. Males, on the other hand, they are XY, so they are either normal vision or they can be colorblind. So I think you're already seeing that because females carry two X chromosomes, they can be homozygous or they can be heterozygous, but males can't. Males are hemizygous. They only have one X. They will never be homozygous or heterozygous, unless, of course, they should get an extra X and have Klinefelters, right? Like the case of the cat or the Klinefelters we discussed earlier in meiosis. All right, so if we were to do one cross, let's say that we had a normal vision mom crossed with, let's say, a colorblind dad. And let's say that from this union, they had a daughter, and the daughter was colorblind. All right, so how's that possible? Okay, so if the F1 daughter is colorblind, then what has to be her genotype? I think we would agree that it's this. So she must be X little n, X little n, right? And we can look at the father and we can see, okay, well, some of that makes sense. He's colorblind, so he would be X little n with a Y chromosome. And the mom, we know that she's got normal vision, right? She's two X's because she's female. And she's described as having normal vision, so she's got a capital N. But how do we make sense of the fact that there's a daughter with two bad N's if dad gave only one of those there? Where'd the other one have to come from? It must have come from mom, so mom must have been a carrier herself. And that's the way we could have figured that out without using a Punnett square, just taking the information given, right? This information here and here. You can just work it out and piece it together to determine that the other little x, or the other little n I meant to say, came from mom. All right, so for this to work, or for you to understand this, you have to get your genotypes down, ensure that you're using X's for females and XY for males. All right, here's a swatch showing how we might determine if a person is colorblind. A person that's colorblind can distinguish that red and green. They would just see gray or the same hue, and they wouldn't be able to see the 7. And in this image right here, I'm showing what's called a pedigree. And you should know that a pedigree is just a system of symbols that we use to track genes in a family over generations, or to track them in a population, like a small population over like a village. We can use these symbols. The circles represent females, the boxes represent males. Fully shading in something means that you're homozygous, you have the condition. Half shading means you're a carrier, right? And in this case, you're looking at a sex-linked example of a pedigree. This is the case of hemophilia. This is that clotting disorder. So it begins with Queen Victoria, who's a carrier. She is XX. Big H, she's normal, but carries hemophilia. And then there's Albert. Albert is XY, and he is not shaded at all, so he's totally normal in clotting. However, they both have a daughter, Alice, so he gave a good X to Alice. There it is. But she's half shaded. That means that she's a carrier. She got a bad X from mom. And then Alice marries Louis, who is not shaded at all, so that means that he had a good X and a Y. And then they had Alexandra, and Louis gave her a good X, but mom gave Alexandra her bad X. So see, that bad X has been making its way through 3 generations. And eventually she marries the last Russian Tsar, who has the ability to clot. He gives their son, Alexis, the Y chromosome, but mom gives Alexis the bad X. See, the bad X has been making its way through multiple generations, lands on poor Alexis, and now he has hemophilia. See how it affects males more than it does females. So that's another characteristic of sex-linked genetic disorders. More people in the population are males with color blindness, are males with hemophilia, are males with muscular dystrophy, and that's because males only have one X, and if it's a bad X, you've got the disorder. But in the case of females, if you have a bad X, you can also have a good one that rescues you. See, they're heterozygous. Recessive alleles are hiding among normal vision moms. But males can't hide it. They either have it or they don't. All right, these are images of dwarfism. Dwarfism is a dominant condition, not found on sex chromosomes or anything, but it's leading us to a discussion on recessive versus dominant disorders. Why is it that dominant disorders are rare, and why is it that recessive disorders are way more common? Here's dwarfism here as well. The reason is twofold. There's 2 answers to this question. Recessive alleles are hiding among normal individuals. So they would be normal / healthy individuals. So that's one half of the answer of why recessive alleles are more common than dominant ones. The other half, number 2, is that dominant alleles tend to be lethal in the homozygous condition. So if you have dwarfism, you're big D, little d. But if you were to inherit 2 dominant alleles, that's lethal. And that removes dominant alleles from a population because they basically are eliminated with the death of a fetus that doesn't develop properly with this dwarfism. And also, like, if you do have the dominant condition, you're showing it right away, right? And you're living the life of a dwarf, and there's, some challenges for these members of our population. So it's a 2-part answer. The reason why there are way more recessive disorders is because they are hiding among individuals that are carriers. Just like we saw here, Queen Victoria, Alice, Alexandra, they're all carriers. They're all X, big H, X, little h. They have normal clotting, but they don't know that they're carrying the hemophilia allele. That recessive allele is hiding among their normal lives. Here, this mom has normal vision, and she doesn't know that she's carrying a recessive allele for color blindness. So recessive alleles are hiding among healthy, normal individuals that are carriers. But dominant disorders, they show up right away. Okay, so know that. This is another example of a pedigree showing, again, the symbol logic of how a disorder might be transmitted through various generations. The boxes are males, the circles are females. Fully shaded means that you have the condition, half shaded means you're a carrier. So if we were to follow this trend, we would have shade here, half shade there, because these are carriers, right? Mom's a carrier. All right, I just want you to know what a pedigree is. A system of symbols that we use to track genes in a family or through multiple generations like you're seeing here, or in a village if we have multiple families that we're comparing. Here's another pedigree showing Widow's Peak. I'll give you this slide, and then if you click on the next slide, you'll see the answers. Now in this case, the Widow's Peak is dominant, right? So there's that there. So if we go back, you'll have that information and some shading that'll help you kind of go through this. Remember, it's dominant, right? Okay. How do we test for genetic disorders? Well, I want you to know about these two. They're called amniocentesis and chorionic villus sampling. Amniocentesis is just collecting amniotic fluid because it contains cells from the baby, whereas chorionic villus sampling is taking some bits of tissue from the placenta. And because the placenta is made by the baby, it also has DNA of the baby. And then from here, what we would do would be this karyotyping that you should remember we talked about some time ago. That's an image of your chromosomes. We can also screen for particular genes. So we can do a variety of things. We can test for a gene, do you have it or not, like Tay-Sachs. And we can also see, do you have too many chromosomes, not enough, are they too short, are they long, right? These are all disorders that we discussed as errors in meiosis during prophase 1, if you have a deletion, a duplication, or that happen in anaphase 1 or 2 when you violate the loss segregation, right? You could potentially have too many chromosomes, like something like that. Okay, so we're going to stop it here. This is completing now the genetics lectures. I'm going to start preparing the lectures on DNA.